in everybody. One too many. And we start with this question of how does a single cell know how to build an entire human? So we're looking at the information in us that determines that, and like what actually contains those instructions. So what determines your eye color or your hair texture, your height, all those. So as we're doing this, we're going to kind of look at DNA as sort of a recipe book where it contains that information and has the instructions on how to build an organism as well as maintain it. It doesn't just stop when the organism's grown, but it keeps regulating how all those things work. So my goal for you guys as we're going through this is that you'll be able to explain that structure of DNA, so how it's made, and the components that make it up. Know a little bit about the scientists who actually discovered DNA, and be able to explain how DNA stores genetic information. So not just like what the DNA is, but how it's working. And when we're done with this, you should be able to model that double helix structure DNA is known for. So for starters, when we're talking about DNA, we're talking about deoxyribonucleic acid. So this is uh, a combination of deoxyribose, a sugar, and nucleic acid, and they bond together and they make these DNA molecules. This is going to be the basic molecule that contains genetic instructions. So this is what organisms look to for development, how things should function, when and how to grow and reproduce. And then DNA is going to be found in nearly every cell of your body. Um, and all that DNA in those cells will also be identical. So all that information, even though it might be using different parts of the DNA, it will all be the same. And you have a ridiculous amount. Your DNA stretched out, uncoiled, stretched out, would be about 6 feet long. All the DNA, and that's per cell, sorry. So if we took all the DNA from all the cells in your body, it's enough to reach from the sun to Pluto. So just getting into a brief timeline of uh, DNA history here. We start out 1869 with Friedrich Miescher, who discovered what he called nuclein in white blood cells. That's what we would call DNA today. Remember, this is only like a couple decades after Robert Hooke first discovered cells. And at this point, we actually have scientists who are able to look inside cells and they're discovering more about them. Then we go from 1869 to 1920s, where scientists now have figured out that DNA has 4 different bases in it. And we'll go through those 4 different bases in a little bit. We jump ahead a couple decades and we have Oswald Avery, who is able to demonstrate for the first time that DNA itself is what carries genetic information. Before Oswald's discovery, they thought maybe protein was what actually stored all this information. There were a lot of ideas for years, even centuries beforehand, about how traits got passed down. People recognized inheritance. They saw like, oh, well, you have your father's eyes or your mom's hair. They knew these things got passed down, but they weren't sure how. And scientists started to think maybe it was protein because it's very closely related to DNA. Oswald was the first one to be able to show that it was actually the DNA that carried all that information. And then we go into the 50s with Rosalind Franklin, who was able to X-ray DNA. James Watson and Francis Crick, you'll see their names quite a bit with DNA, were the first ones to show that double helix model. And then Watson Crick and Maurice Wilkins ended up getting a Nobel Prize for their research into DNA. When we're talking about DNA, we also have to talk about how it's structured, what it's made up of. So, DNA is these repeating units called nucleotides. Each nucleotide has a phosphate. We have a phosphate. Let's see if my. Nope. We have that phosphate. We have a deoxyribose. That's the pentagon shaped thing here. And then we have a nitrogenous base. And there's those 4 different bases, the adenine, thymine, cytosine, guanine. So a base, a sugar, and that phosphate make up the nucleotide, and then those nucleotides are going to bond together to actually form the DNA strands. That's our 4 DNA bases again: adenine, thymine, guanine, and cytosine. When we're talking about these in class, I do expect you to know those names, those complete names, but a lot of times we'll just refer to them by their letter, the A, T, G, or C, just because when we keep going through all those, kind of gets to be a mouthful repeating adenine, thymine, guanine, cytosine. But these will form sequences that make a code, and that code will actually tell cells how to make proteins. That code also is what will determine the different traits and characteristics about an organism. So that's where our hair color, eye color comes in. There are specific pairing rules with it, though, where the adenine always pairs with thymine, cytosine always pairs with guanine. It doesn't matter which side of the double helix the base pair is on, it's still going to pair with the other, and they appear on both sides. Easy way to remember these is apple in the tree. So adenine and thymine, A and T. So apple in the tree, and then car in the garage, cytosine and guanine, C and G. So apple in the tree, car in the garage. So they will always pair together. Also, remember they pair together with hydrogens in the middle. AT gets 2 hydrogens. CG has 3 hydrogen bonds. So when they pair together, they're going to end up looking like this. Like here we have a G and a C. There's T and A or AT. So remember how I mentioned that we'll have those pairs on either side. So your nitrogenous base can be on either side. The phosphate and the sugar, that deoxyribose, form the sides of the ladder, and then our base pairs will make the rungs of the ladder. Normally, the double helix gets referred to as like a twisted ladder or spiral staircase because that's what the structure looks like. It also runs what we call anti-parallel. This is kind of like cars on the road, where the cars are running side by side each other, but they're going in different directions. We don't want those cars to run into each other, of course. So same thing with our DNA strands. They're going to run side by side, without running into each other, and they'll run in opposite directions. So one strand goes 5 to 3, the other goes 3 to 5, which we'll get into later. Now, going back for a second, Rosalind Franklin was actually the first person to successfully X-ray and identify what that structure looks like. She was an X-ray crystallographer, um, meaning she specialized in X-raying crystal structures, so things like diamonds or quartz, trying to look at how their atoms are arranged. So she did a series of X-rays looking at DNA. She got interested in this and tried to X-ray cells to capture their DNA. And photo 51, that's what's shown over here on the right, is probably her most famous one that shows that DNA structure. Her work, these X-rays, ended up being shared with Watson and Crick, who used them to further their research. Rosalind Franklin was not aware that Watson and Crick were using those, or that they had been shared with them. So Rosalind Franklin didn't really get any acknowledgement for her work until well after she died. Part of the reason she didn't get a Nobel Prize for it, she didn't know about it, and they don't give Nobel Prizes posthumously, so it was too late. But her work at least let them identify that structure. And Watson and Crick used it, used those X-rays, and correctly identified that DNA is in that double helix structure. They were able to identify that those bases, those nitrogenous bases, come in complementary pairs, that those strands run anti-parallel, so opposite directions, and they were able to even identify those dimensions of the double helix. I kind of like this quote from them. The structure has novel features which are of considerable biological interest. Considering how important DNA has been in biology and medicine research, and they're like, yeah, we think it'll be important, but they seemed very restrained about it. So um, all those bases end up coding for proteins, ultimately. Now, those individual bases or those pairs don't really do much on their own. It's only when they start forming segments or sequences of DNA that they'll code for proteins. So we call those segments genes. Those genes also determine traits, but a lot of what we'll look at is how they code for proteins, because those proteins work as like hormones and enzymes. They're going to regulate bodily functions. They tell your body when to grow. They tell it what to do and when. Now, a complete set of your DNA or genes in your body is called a genome, Genome, genome. Your genome is made up of about 20 to 25,000 genes, or 3 billion base pairs. So that's 3 billion of those ATGC pairs. The double helix structure has some advantages to it, where it allows for the DNA strands to be coiled up compact, like kind of like they're twisted and smushed so that they're able to fit inside the nucleus. So, despite the fact that there's about 6 feet of DNA in each nucleus, um, it basically it winds up really, really tight and small. There's also an advantage with um, the sugar phosphate backbone forming those sides of it that gives some stability to the structure. And the fact that it comes in those complementary bases also means it's like there's backup information there because if like one side of that double helix gets damaged, the other side, if it's still there and intact, still stores the same information. We know that, or your cell would know that uh, it just needs to replace it with the correct corresponding nucleotides. That also leads us into how DNA replication works. So, this is what was happening during that S stage and interphase when we were learning about the cell cycle, and DNA needed to be copied before the cells could split. It did this DNA replication. So, an enzyme goes through that double helix and splits it or unzips it right down the middle. It breaks those hydrogen bonds and separates out uh, the DNA into two separate strands. After they're separated like that, nucleotides will be paired back up with them. So, an exposed adenine would get paired with a thymine nucleotide, or a cytosine would get paired with a guanine. So that when that DNA replication process is done, there's going to be two identical sets of DNA. Each of those DNA molecules then will each contain one of the original strands, because remember that strand split in half, and then they'll each also contain one new strand from those nucleotides that were matched up with it. As assuming everything goes right. Now, there are possibilities that the DNA strand either isn't replicated correctly or changes can happen in it. And we call those mutations. So that's any of those like mistakes during DNA replication. This could be damage caused to the DNA by outside things like radiation or chemicals, or even by viruses, because viruses will implant their DNA into a cell for the cell to make more viruses, which was the case with Henrietta Lacks. Or she contracted cervical cancer, that HPV had inserted its DNA into her cells, which changed how those cells um, grew. Now, I think mutations tend to get kind of a bad rap. They normally sound like they're always going to be harmful. It sounds like a negative thing, but mutations can also be beneficial. This is how we've ended up with so much variety of life. Um, it drives all these different traits and genetic variation that we have, where we end up with a different eye color, different hair color, because there were mutations in those genes, and it gave some kind of advantage to an organism that ended up with them. There is also the possibility with mutations that it won't have any effect at all. We'll be doing um, DNA translation and transcription, where we're going to look at how it becomes RNA and codes for proteins. And it's possible that that DNA can change and still make the same proteins, and it won't change anything at all. Now, I know a lot of this sounds like, oh, we're learning about old dead scientists and this vague idea of, oh, here's DNA and it codes for what we look like. Why does it matter? Which is fair, but um, DNA is going to be increasingly important, especially in medicine. But we also use it today for sequencing and fingerprinting, where we can use it to identify individuals. I always like it in crime shows because that's like the go-to. Oh, we found the DNA at the crime scene. We'll see if it matches the suspect, which is real world. We can match people's DNA. We can see if it's an exact match or if it would identify a relative. Like I've done an at-home DNA kit that also showed me uh, relatives, sometimes second, third cousins that I never knew about. But the other side of this is what we can do with it down the road. So it's not just identifying people right now, but we can test for specific genes that might cause diseases like cancer. And the big hope is that we'll be able to use gene therapy and CRISPR technology to alter that DNA, like change those genes to either fix them or even maybe like replace or remove um, mutated genes to treat those diseases. Or we might be able to at least identify that someone is likely to have a certain type of cancer because they have that gene. All right. You guys have any questions on anything?